Again, welcome and thanks for joining us today. This is our TrustML team. We've been running this initiative for around a year very soon now. And all of us, we have a new member, Hao Han, who's joined us recently. To talk a little bit more about TrustML, here are our goals and efforts. We have a curated list of tutorials, courses, papers, and some more resources on our website for easy access. We every now and then do a Twitter highlight series for budding researchers, and we also have Rising Star Spotlight talks for young researchers. We have an active Twitter handle where we have a lot of discussion and debate going on. And very soon we will organize a symposium, so please stay tuned for that. Talking about today's agenda, from 12 to 12:45 p.m. Eastern Time, we'll have Marinka's talk. Please feel free to submit, upvote, and comment on questions using the Zoom Q&A tool. From 12:45 to 1 p.m., we'll have a Q&A session and a moderated chat with Marinka. So here I'll ask some technical questions and also some questions specific to the speaker's journey in ML. After that, we'll have a 5-minute break, and at 1:45 p.m., at 1:05 p.m., we'll have a participant-driven discussion, which will not be recorded or live streamed on YouTube. So talking about today's speaker, we have with us Marinka Zitnik. Marinka is an assistant professor at Harvard University with appointments in the Department of Biomedical Informatics, Broad Institute of MIT, and Harvard. Dr. Zitnik investigates machine learning, focusing on challenges brought forward by interconnected data, so basically heterogeneous data in science, medicine, and health. Her research has won many awards, including Best Paper and Research Awards from the International Society for Computational Biology, the Bayer Early Excellence in Science Award, Amazon Faculty Research Award, a Rising Star Award in EECS, and a Next Generation Recognition in Biomedicine Award. And she is the only young scientist who received such recognition in both EECS and biomedicine. Today, Marinka will talk about graph representation learning for biomedical discovery. Marinka, the floor is yours.
Thank you so much for this nice introduction, and thank you to the organizers for inviting me today. I'm really delighted to be here and to talk about graph representation learning for biomedical discovery. Before I start, let me share my screen. Okay. So I hope you can see my slides now. So let's begin. I will talk today about graph representation learning for biomedical discovery. And the first question that I would like to address is to really elucidate a bit the challenges and aspects of biomedical data that make biomedical data unique and different from different modalities, and then also present exciting challenges and opportunities for algorithmic innovation. So for this talk, I'd like to emphasize a couple of these unique aspects of biomedical data. The first is that for most phenomena that we are modeling in biology and medicine, we really need to integrate heterogeneous, confounded data that can span from molecules all the way to the level of society and populations. And that data typically does not fit into a large single flat matrix-based data representation, because the phenomena are typically so complicated that they require us to develop approaches that can integrate information about genes, their interactions between them, and molecules at the level of cells, all the way to behaviors. And so the data is multi-scale in that sense. It is heterogeneous in the form that it comes in forms of experimental readouts, curated annotations, self-reported measurements, and also confounded in different ways because it's generated by different technologies and measurement platforms. The second point is that in order to really enable biomedical discovery, it is important to be able to translate predictions into what I will call actionable hypotheses here. And by that, I mean really that it's important to have predictions that lend themselves immediately to testable hypotheses that can be then tested experimentally in downstream experiments in wet lab biological laboratories or in clinical settings. And so that, of course, means that accuracy alone is no longer sufficient criteria for many of machine learning models, because there are other aspects that are incredibly important, including those that including explanations for predictions made by models that can reveal previously unknown biology. And that's something that I will also demonstrate in the rest of today's talk. The second important point for today's presentation is that for problems at the frontier of science, there is typically very little label data available. And so why is that important? That's important because recent breakthroughs in AI have typically depended on having large and massive data sets and some means of and some form of labels, even though that labels might be noisy or weak, but there is typically some notion of labels existing. That is not the case for problems that are really at the frontier of science. For example, when one wants to develop a drug for a disease we have never seen before. Of course, there will be no labeled examples representing successful treatments for that disease that is just new disease. And so that makes biomedical data quite unique and presents several challenges that need to be addressed that I will highlight in today's presentation. The approach that our group is taking is that we're addressing problems around heterogeneous and multi-scale data by typically representing data in the form of rich, complex network-based representations or graphs. And so that often means that we construct large networks of biological or biomedical entities, and these biomedical networks and graphs really pervade biology at the level of interactions between proteins and molecules within a cell, to the level of interactions between various diseases that might coexist within an individual, and all the way to interactions between people in a society or local community. And so we see these networks at all different scales, and it's important to consider them in order to enable predictions. Now, of course, when we have these graph-based representations, a natural question emerges, which is how do we learn and reason over those representations? And so that is one of the main focus research areas of my group, where we are developing methods for graph representation learning that can learn and reason over this rich network-based representation. There has been a tremendous surge of interest in leveraging graph representation learning methods over the last few years in order to learn meaningful representations of biology. And we have a survey paper on arXiv, and it's currently in review, that emphasizes and describes some of the efforts that have been enabled by graph representation learning in biology and medicine. A prevailing approach for graph representation learning are graph neural networks. So what are graph neural networks? In a single slide, and without going too much into detail, the idea is that we start with a network that I'm showing a simple example here on the left part of the slide. And of course, that network can be complicated in the form that it might have multiple different types of nodes, different semantic relationships, might be dynamic or multi-scale, but in the simplest case, it's a very simple connected graph. And so graph neural network is then a nonlinear deep transformation function f, and that function specifies how each and every node in the input graph maps to some d-dimensional embedding. And so that d-dimensional embedding is a representation of the node in the embedding space. And so the ideal graph neural network architectures are then to optimize the structure of the embedding space such that performing linear algebra operations on vectors in the embedding space will reflect topology of the input graph. Now, there are many ways how to optimize embedding spaces, and so that has given rise to an entire field of research where there are many neural network architectures have been developed over the last few years. Behind all those methods, the intuition is that the goal of function F is to map nodes to the d-dimensional embeddings such that nodes U and V that have similar connectivity and local network neighborhoods on the left will be represented by points that are close in the embedding space. And that is achieved by these functions F typically have a form of neural message passing operators that are mechanisms that represent trainable operators, and those trainable parameters in those operators specify what is the most useful way to propagate embeddings along edges of the graph such that those embeddings are indicative of the structure of the graph, so can predict the structure well. So this is the self-supervised learning perspective here. So with that background in mind, the plan for today is that I would like to cover two topics. And so the first topic that I'd like to cover is to explain a bit and describe our recent effort on developing graph representation learning methods and graph explainability methods and how we use them to support a large national effort in finding cures for an emerging disease. And so the second topic that I will briefly mention is more of a highlight of a collection of AI-ready data sets for drug discovery and development that is really targeted at machine learning community in order to enable more of machine learning researchers to work in these exciting fields of biomedical discovery. So with that in mind, let's start with the first topic. And so the key challenge facing biological discovery is really this observation that generalizing to new phenomena is incredibly hard. But typically that is where most exciting opportunities are. And so why is that so hard? Because prevailing graph neural network methods require abundant label information. Labeled examples are typically very scarce. That is indeed the case across many different problems. For example, when one would want to develop methods that could diagnose patients that are otherwise very hard to diagnose, to identify and model diseases that are rare, to find treatments for new emerging diseases, new pathogens, to answer questions about novel drugs in development. The core of this challenge is that prevailing methods assume that the input graphs looks like the one I'm showing on the left part of the slide. So we have a graph, and most of nodes in that graph are already labeled. We know what the roles of those nodes are. And so given that information, the goal is then to predict the labels of remaining nodes, if we think of node classification problem. But what really happens in real world is that we get a picture like the one on the right, is that most of the graph is unknown. It's kind of dark matter. There's very few labels that one can have. And so how can those labels be effectively leveraged to make prediction? And so the goal is then that we will spend the next few minutes in is to try to unpack and describe an approach that can realize the goal of how to make predictions in a new graph or for a new set of labels when we only have a handful of labeled examples for that task. And so one slide of background here that many might already be familiar with on this call, but I'd like to introduce because of relevant terminology, is of course the field of research on meta-learning and a particular instantiation of meta-learning in the case of supervised learning, which is known as few-shot learning. And so here in this background slide, I'm showing an example of how few-shot learning is typically achieved in area of computer vision. And so here a particular simple example that I'm showing is the one on image classification. And the way that would operate, the way this problem is addressed in the few-shot learning setting is that the strategy is to have to train models by in two phases. The first phase is known as meta-training phase. This is a phase where a model will be presented with a large number of training tasks, and each task is designed such that it's really challenging for the model, in the sense that for each task, there are just a few examples for each of the classes, for each label. So in training task one, we have two labels for each class. So 3 classes and 2 labels for each of that class. And so the model would be trained on this small, tiny data set in order then to answer in such and be optimized to then answer question on the query set. And so this would be repeated many, many times for different subsamples of the training data. And so the model would be primed to solve these training tasks effectively, so that at test time, when we would want to build a duck versus dolphin versus chicken classifier, the model would be able to quickly transfer that useful knowledge and quickly adapt to the new set of 3 classes that have never been seen in the meta training phase, and be able to distinguish ducks versus dolphins and chickens, having only 2 labeled examples for each class. And so the setup here the example that I'm showing here is that of 2-shot 3-way image classification. Why? Because for each we have 2 labeled examples for each class. And so this strategy has been explored quite thoroughly in the context of images. But what our goal here is really how to develop strategies that can enable meta learning for graphs. And I will describe an approach that we have developed for that. And so this approach, which we published at NeurIPS a few months ago, is known as GMeta algorithm. It's a very general approach that can work in a variety of different settings in graph representation learning, including when we have data sets consisting of single graphs or multiple graphs. But for brevity, I'm showing here the simplest possible situation, which is that of a setup where we have a single graph G, and the task is to predict labels of nodes in that graph. Imagine we have 2 types of labels, yellow labels, and then different shades of yellow, and another label set represent different shades of blue. And so the setup here would be that we need to have a graph neural network-based meta learner that will be able to in the meta training phase, be considering only label the yellow label set, and then be able to very quickly adapt at test time to the new blue label set and make prediction for this node B, having only essentially 1 example of node that has a blue label. And so that is something that is challenging in this particular case. It would correspond to a 1-shot and 2-way few-shot learning problem if we assume that we have just 2 shades of yellow and 2 shades of blue. So the idea of GMeta overview is that the overall overview is similar to general few-shot learning settings, so that we again have these 2 stages the first stage is the meta training phase, and the second is the meta testing phase. In the meta training phase, we will train a graph neural network in such a way that we will define a number of different training tasks, and each task will be defined on a different set of labels. And then the GNN model will be asked to make predictions looking only at a small number of labeled nodes for each class in the label set. And now during the meta test phase, a new label set will be presented. And all these labels will be sampled from the same graph. So the key idea of how to then really train a graph neural network, so how to optimize those parameters that specify how neural messages are propagated along edges of the graph, is the idea of leveraging local subgraphs. What I mean by that is the following. So each task, both those in meta training phase as well as in the meta test phase, is defined as a distribution over subgraphs of the graph that defines the task. And so each subgraph is defined as a local enclosing subgraph around a particular node for which we have the label. And so the idea is then to use those subgraphs in order to learn a global set of parameters for a GNN, and then use those global set of parameters as a mechanism to transfer knowledge from one task to the other. So each task is then described by a subgraph signature function. And that subgraph signature function is a function that maps the structure of and patterns found in the enclosing subgraphs of labeled examples to effectively initialize a GNN for a new task. And so that additional information that we bring in by looking at enclosing graphs allows us to effectively prime GNN models. It is possible to theoretically analyze this approach, in particular the motivation for why looking only at local subgraphs around labeled nodes is sufficient. So I'm not going into details here, but the idea is that in graphs and in network science in particular, which is an area that has been developing for the last several decades, it has been shown over and over again that typically the most informative information for predicting labels of nodes is found in the immediate local vicinity of the target node. And so it's possible to formalize that and show that the influence that a particular node might have on the target node that we want to make prediction for really decays exponentially as we go further away from the target. The implication of that is that these local enclosing subgraphs around target nodes contain all the relevant information and preserve near the same feature information as looking for the entire graph. And so to give a bit more intuition behind that is that we can adopt the following reasoning. So when we think of graph representation learning and graph neural networks in particular, we typically are considering two sources of information that is leveraged to really increase and predictive power of these models. And so one source of information is label propagation. We assume that nodes with similar labels will be close to each other in the network. Therefore, it makes lots of sense to propagate that information effectively. The other assumption is structural similarity. Nodes that might actually be quite far away from each other in the graph, they might have similar network shapes, similar motifs and structure present in their local neighborhoods. So that can also inform prediction of labels for them. Now, when labels are scarce, as in the case of our few-shot learning setting, label propagation is not sufficient. It's simply not possible to reliably propagate a label and diffuse labels along edges of the graphs, because we have so few labels that we cannot do that in any reliable manner. And so because of that, we want to better leverage structural similarity. And that is exactly what local subgraphs can do. So to recap, GMeta is a general. It can be used for any kind of graph machine learning task. And I've been focusing primarily on node classification tasks, but it also applies to other problems. It uses local subgraphs to specify the mechanism that transfers information across graph and across label sets. This allows the approach to learn how to quickly adapt to a new task using only a handful of labeled examples, and it's also theoretically justified, as we showed that the evidence for a prediction can be found in the local subgraph surrounding the target node or edge. And so with that in mind, I will now go into a second part of this topic, which is how to use these techniques and apply them for problems in drug discovery and development. And so a bit of context for that, since I assumed some of the call might not be familiar with drug discovery development process. Drug discovery is concerned with the design and development of a new drug from scratch and delivery of the drug to the market so that the drug can be used by patients and it's safe and effective. This process is incredibly resource intensive and expensive. On average, it takes 12 to 16 years and it costs between $1 to $2 billion to design a new drug from scratch. And this process consists of several stages or phases. that go from first identifying what or which out of millions of possible compounds are those that might possibly have certain properties that are required for the drug in step one, and then filtering those drugs to those candidate compounds through in the following stages of preclinical research and clinical research to make sure that those compounds are first safe in animal models and then safe also in human patients, and also that they are effective, really treat the disease as intended. And then finally, if all this is process is successful, then and luckily one compound will reach the stage that when it can get approved by FDA and then used by patients and put on the market. And so the reason why this process is so long and expensive is because it's of incredibly high failure rate of compounds that initially seem very promising and have certain properties that are required of a drug, but then very quickly turn out that, well, they actually cannot be transferred effectively to humans from animals, and then they are not safe, and so on. So that is the key reason why the process is so expensive and takes so long time to develop a new drug from scratch. So, and why is that particularly challenging? And I'm mentioning the length and the intensiveness of this process is because this traditional iterative approach of iterative development and experimental and clinical testing, approval of new drugs, is not feasible in many situations. And so in the rest of today's talk, I will focus on one particular situation and the project that we did, and that was that related to COVID-19. And that is an example where rapid therapeutic innovation was required. One could not wait for 12 to 16 years to find or identify what drugs might effectively might be effective for COVID-19 patients and likely have some therapeutic effect. The question really is how to do that rapidly, and that has motivated the project that I'll be mentioning. And so that the project that my group was part of and is a national COVID-19 task force that was centered here in Boston. And the work that we did between March 23rd of last year and actually June of 15 is work in less than 3 months, in which a team of machine learning scientists and our collaborators, biologists, virologists, were able to compress amount of work that typically requires years or months to do in really days or weeks. And the way this was possible is through the use of algorithms that I just described and the use of explanations of predictions made by those algorithms. And so in the next few minutes, I will describe this process a bit in detail. So the key question that we were tasked with in March last year was to design a model that can predict what drug will treat what disease. So think of it as a problem where we have a large bipartite graph. One part is a part that consists of all drugs that we currently know of. So those were thousands of drugs that are approved currently in the US market. And on the right, we have tens of thousands of common diseases, and also we have this new disease that we haven't seen before, and that was COVID-19 disease. The question then becomes, if we take into consideration what are the diseases that existing drugs treat, our goal is to predict what drugs that are on the market might treat this new disease that we haven't seen before. So formally, we would think of it as a link prediction problem on this bipartite graph. Why is it challenging? It's challenging because COVID-19 was a never before seen disease. So we don't have any label for it in the sense of successful drug that already works that one could use to train the model in order to find more drugs that could possibly work. right? So it was really like, can we find, for the first time, can we prioritize and identify what are those drugs that might likely have therapeutic effects, which really goes back to that few shot learning problems I was describing before. The first question that emerges is, how do we represent COVID-19 in a way that will be amenable to machine learning analysis? And so for that, we relied on the data generated by Gordon et al. From UCSF. And what they did is they took the virus and they expressed 26 out of 29 SARS-CoV-2 proteins. Then they used a technique known as mass spectrometry to identify 300 proteins in human cells to which those viral proteins bind. And so those 332 human proteins that are attacked by the virus were used in our project to define viral disease module. So COVID-19 was then represented as a subgraph of the human protein interaction network. That subgraph was a subgraph where nodes were proteins in humans that the virus attacks, and edges in the COVID-19 disease module indicate interactions between those human proteins in human body. So that was COVID-19 disease module. And then we went and represent each and every drug that currently exists in the US market, and represent every drug as another subgraph of nodes that the drug targets. So that would be a drug module. Each drug module is a subgraph of protein interaction network. Its nodes are proteins that the drug targets. We say that it binds to those proteins in order to change their activity and treat the disease. And with that data in mind, we carry out experiments in the following manner. So we took the COVID-19 disease module and supplemented with information about large number of other common diseases, and we did the same, use a similar approach to represent each and every approved drug as a subgraph of proteins to which the drug binds. All that information was overlaid on the human protein interaction network. So the way to think of it is we have this underlying graph, which was a graph where nodes are human proteins, edges are indicate information how proteins in human cells interact with each other. And then on top of that graph, we overlay many subgraphs. And so subgraphs represent either diseases or they represent drugs. And so that was the repurposing knowledge graph that we constructed. And the goal then was, given information about many other diseases, tens of thousands of them, and existing treatments for them, how can we use that information to specify and use that information during the pre-training phase, such that in the meta test phase, we will be able to quickly optimize the model to predict candidate treatments for COVID-19, right? And so the challenge here is, again, we have no examples of successful treatments for COVID-19 and no labeled examples to train the model. So it was important to have information about successful treatments for other common diseases and use that information in the meta training phase of the model. And so the approach or the instantiation of the GMeta, one of the variants of GMeta algorithm I've been describing before is the following: that in the meta training phase, we define a large number of training tasks. Each is defined around one disease and a small number of successful treatments for that disease. And then in the meta testing phase, we are presented with this emerging pathogen that one needs to make prediction for in terms of predicting links between drugs and diseases that are indicative of treatment relationships. So let me talk a bit about results. When we first implemented the model, we wanted to understand what is the potential and possible use and utility of that model. And so to that extent, we tested in April 2020, the accuracy of predictions returned by GNN methods, as well as other computational methods for drug repurposing against drugs that were in clinical trials at the time. So the idea was to obtain a ranked list of drugs that were returned by predictive model, and then check whether drugs that were at that time in clinical trials were at the top of that ranked list. That would indicate that the model, at least in principle, can identify what are drugs that research community thinks might be promising for COVID-19. And so in this AROC plot, we can see that GNN methods really outperform many alternative methods that has then motivate us to use GNNs to generate a final set of predictions that we then gave to collaborators who did experiments in the lab. And so our collaborators from National Emerging Infectious Disease Laboratories took predicted ranked lists of drugs produced by our computational strategy for COVID-19 drug repurposing, and they in a laboratory with live virus carried out an experiments in which they have cell cultures with where they first put African monkey cells and drugs that were predicted by our model, and then measure what happens to those cells if cells are infected with the virus or not, and then repeated this process for human cells and in a particular cell culture with human cells, the drugs in different dosages that were predicted by our model, and then also measure whether cells survive when they are infected by the virus and when the drugs are there, or whether cells don't survive. And so they screened 918 compounds for this efficacy against SARS-CoV-2 virus, and results were incredibly satisfying. So by screening this, by carrying out these experimental screens, we obtained a 62% success rate when doing the screening according to the ranked prioritized list of drugs written by models, which is in sharp contrast to 0.8% hit rate, which is what would be possible without using a machine learning approach here. And so that's an order of magnitude better hit rate than alternative approaches would offer. And so working with biologists and virologists very closely over such short period of time, there are several takeaway messages that I'd like to share that I think it might inform a machine learning community. So the first is certainly that it's still the case that the problem of zero-shot learning is incredibly challenging. And so it's not solved. There are so many open questions here about how to really develop models that can generalize to a new disease, in this case, that we haven't seen before, and make useful predictions about that disease. And most prevailing approaches cannot do that, and that are published in literature, and are also typically developed and evaluated in easy settings. So it's very easy to find another drug for a disease that already has been explored so much in terms of making predictions for that disease. Second is, it became really important that users, in our case, domain experts, were able to interact with the ML model somehow and provide feedback for that ML loop. That was crucial to be able to really rapidly iterate on model development and deployment, and really make sure that domain experts trust our predictions. Because these downstream experiments that I've been mentioning that have been done in wet lab laboratory, those are resource intensive and expensive experiments. So one would not just blindly follow a single one-dimensional prediction or returned by the model together with some confidence scores and just blindly use that information to do downstream work. And so for that, we have been working quite a bit on ways that would allow users to engage with graph neural network models, and that could, in an effort to enable scientists to provide feedback on model development, to build trust in ML predictions, enable scientists to ask what-if questions and receive predictions that can be interpreted meaningfully. And in the context of the project that I've been mentioned, we have used one of the algorithms that we previously developed, known as GNN Explainer, which is an explainability algorithm that can explain prediction in a post hoc tool that can explain predictions made by GNN models. And so the way it operates, its key idea is that it will summarize where in the data, where in the graph the model looks for evidence for its prediction. The way it will do that is by finding a small subgraph that is most influential for a particular prediction. And so the way this is achieved is that we assume that we have our GNN model F, we have a prediction X, and so we have so we have input X for which GNN model F makes prediction. And so this is the input to GNN Explainer. And so the output will be an explanation that is formalized as a small subgraph represented as some form of mask for that input X, together with a small subset of node features. And so that subgraph is found through an optimization, by solving an optimization test that intuitively what it does, it optimizes mask in such a way that a node V would be included in the mask as giving an explanation for prediction X. If by removing V from the graph, the removal would strongly decrease the probability that the model F makes for prediction X. In that case, we would think of F should be included as part of an explanation. So in that algorithm implemented, we were now able to really in a more principled way provide explanations to domain experts here. So when the domain expert asks questions such as this query with a form of state in which is a drug treat hyperlipidemia, and what's the disease treatment mechanism, we can then return not only a score that represents the probability of a link between this drug and disease existing in the graph, but then also find a small subset of genes and functional pathways that according to the model represent treatment mechanisms. And that's much more useful for domain experts to then design experiments for, rather than simply looking at prediction scores. So in the context of the projects that I have mentioned, these techniques have allowed us to analyze what are drugs that were predicted correctly and also confirmed in laboratory experiments. And so we found out that 76 out of 77 drugs that were predicted by the model and confirmed to successfully reduce viral infections are those drugs that the way they work is that they don't directly try to fix the proteins that the virus attacks. Instead, those drugs rely on network-based action. So the way they operate is when a patient takes the drugs change behavior of some proteins that are actually far away from those proteins that the virus attacks. And those proteins in turn interact with some other proteins who eventually are targeted by the virus. So the way this was possible to discover is by really looking at lots of these explanatory subgraphs returned by the model and start to try to figure out what is common to explanatory subgraphs for drugs that were successful in inhibiting virus versus drugs that were not successful in doing that. And so we now call this class of drugs as network drugs because they operate through this network-based action. And it also shows how they could not really be identified by alternative strategies. So I'm already running over time, so I will have one final slide, and this actually concludes my first part. And the second part, I will have one slide on a new initiative that we founded around half a year ago. It's known as Therapeutics Data Commons. So Therapeutics Data Commons is an open science initiative. It's available at the website tdcommons.ai. And it's really an initiative founded by a number of collaborators in the CS and ML space in an effort to provide AI-ready data sets and meaningful formulations of tasks, such a way that together with an ecosystem of data loaders, model evaluators, strategies for data set split, leaderboards, and so on, in order to facilitate algorithmic and scientific advance in this broad area of therapeutics. So what we believe is that there's been the drug discovery and development in particular offers so many opportunities as well as hard challenges and opportunities for impact, but it's often incredibly hard for machine learning scientists to enter these fields, as one needs to have quite a bit of domain knowledge in order to really understand what are problems that one can tackle, where to get data sets that are ready for ML model development and evaluation. And so these are some of the questions that TDC addresses now. So we have a nice website, a paper, GitHub repo. We are currently seeing over 3,000 new active users every month. Those are both scientists from the machine learning community, as well as biomedical research and pharmacology and pharma industry. And there is a team of volunteers and students in my group, as well as in the group of my collaborators who actually maintain and actively develop therapeutics data commons. And so I highly encourage everyone to check it out, to kind of move away from these classic boring benchmark data sets in ML. And so with that, I will stop here. Thank you everyone for listening, and I'm happy to take questions.